Okay, hello everyone. Thank you for the nice introduction, and Jay and Wolfram for the opportunity to present my work here. So I'm coming from Hesham Sadek Lab in UT Southwestern. So today I want to talk about some data that we are developing in a project in the lab related like how glutamine metabolism is helping the heart as a metabolic adaptation during cardiac hypoxia. So as we all here know, the heart has only a transient regenerative potential during the early days of postnatal life, and this is because a switch from hypertrophic growth that allows to regeneration, hyperplastic growth that allows regeneration, to a hypertrophic growth that impairs regeneration. So historically in Hesham Lab, we have been working in this problem, and we're starting wondering like, okay, what's happening in these first days of life that blocks the proliferation of the neonatal cardiomyocytes? So one of the reasons that the hypothesis started with was in organisms that has cardiomyocyte proliferation and regeneration, like zebrafish, or during the embryonic mouse development where the cardiomyocyte proliferate, the difference with the postnatal life in mouse is like there is a mix in between the oxygenated and the non-oxygenated blood. So the oxygen levels that these tissues are exposed to are different. So actually this causes like there is a progressive increase in the mitochondrial mass of the tissue as the mouse transition in the postnatal stage, and also an increase in the mitochondrial ROS productions. This leads to an accumulation of oxidative DNA damage, as you can see here by 8-oxo-G staining, that leads eventually to the activation of DNA damage response, and this is a signal for exit the cell cycle, and that loss of proliferative capacity. How can we relate this with metabolic process in the heart? So, as is also very established in the field, during the neonatal life, there is a transition from glycolytic metabolism to fatty acid oxidation. This increases the DNA damage by increasing the mitochondrial ROS productions. Here we have treated in vitro and RBMs just with fatty acids, and this resulted in an increase in the DNA oxidation, and in the DNA damage signal, just by treating the cells with fatty acids. Also by a fluorescence reporter that allows us to track real time by microscopy the amount of ROS that are being generated or reaching certain cell compartments. By targeting this sensor to the chromatin by fusion with H2AX, we see that the treatment with fatty acids in these cells results in almost induction of 2 or 3 times of ROS abundance in the proximity of the chromatin. Also for models in vivo that has been developed in the lab, we generated a conditional mutant of PTK4. This guy is basically the main that controls in the heart what is entering the mitochondria. Pyruvate derives from glucose or fatty acids. So when we knock out PTK4, there's an increase of the utilization of glucose-derived carbons in the mitochondria instead of fatty acids, resulting in a decrease in the activation of DNA damage response, and subsequently increase of cardiomyocyte proliferation, and also a regenerative potential. Another model that we developed, okay, in this previous model, we were controlling the metabolism directly. However, what happens if we control the oxygen level, the environments that those mice are exposed to? So the lab developed this chronic hypoxia model, and the word chronic is really important. We are not talking about like an acute response of hypoxia, the classic acute response of hypoxia. So we adapt the mouse by dropping the oxygen 1% per day during 2 weeks till we reach 7%. This is more or less the amount of oxygen that we would find in the top of Mount Everest. And we keep the mouse 2 weeks under these oxygen conditions. This results in a reduction of the mitochondrial ROS productions, both at the level of superoxide, but also hydrogen peroxide and other ROS. As expected, this resulted in a decrease in the DNA damage oxidation, and the decrease in the activation of the DNA damage response pathway, and increase in cardiomyocyte proliferation after 2 weeks in hypoxia. So this can be used for regeneration. That was the next step. So keeping the same model, but adding a step of raising again the oxygen level by 2% per day. We don't want to get them directly in normoxia again. That would be like a kind of like a reperfusion shock. Suddenly the tissue that has been adapted to low oxygen is getting a bunch of oxygen back. So this 1-week adaptation allows the heart to adapt back to normoxia. So when we perform MI before the hypoxia treatment because we don't want to precondition the heart to the injury. We want to make the injury and then do the hypoxia treatment. We see that here there is an increase in the ejection fraction, and of course this goes together with a reduction of the scar size and an increase in cardiomyocyte proliferation. We wonder, like, okay, hypoxia has been known for mediate a lot of metabolic adaptations, and also here we're trying to mimic kind of like a return to the neonatal life in terms of oxygen levels. So what's going on with the metabolism in those hearts? So we perform metabolomics after 2 weeks in hypoxia in the ventricular, just in the ventricles of the heart. And we found that just by principal component analysis, we get a complete segregation of both groups just by their metabolomic profiling. We perform a pathway analysis in the differentially expressed metabolites, and we found a lot of differentially regulated pathways. But of course, we had glycolysis as we will be expecting, but the most differentially regulated pathway was glutamine metabolism. So what's the thing that is going on here in hypoxia? Why we depend on glucose? We depend on glucose because now our mitochondria is going to be working wireless. Of course, it's not going to be totally stopped. We are still at 7% oxygen. There is some remaining oxygen, so the mitochondria can still do some things, but of course, it's not going to be able to sustain the ATP demand that a cardiomyocyte needs to be beating all the time. So glycolysis takes over and generates ATP. What's the problem here? Glycolysis is producing NADH in the cytoplasm. Usually this would go to the mitochondria, donate the electrons to the electron transport chains, and we recover the NAD. However, here, mitochondria would not be doing that as efficiently. So usually the cells convert pyruvate into lactate to regenerate NAD and keep the glycolysis going and keep generating ATP levels. However, we found that the levels of lactate in these hearts, in the hypoxia, they weren't changed compared to the normoxia. So we wonder, okay, there is an alternative pathway that is contributing to the NAD plus generation in the cytoplasm that can explain this chronic adaptation during the proliferation induced by the hypoxia. We found that the glutamine, and this correlates with the previous pathway analysis, it was increased in hypoxia compared with the normoxic hearts. And actually, when we treat H9C2 cells in vitro in hypoxia with PBS or with glutamine, glutamine decreased the overall NADH levels in these cells, indicating that glutamine can be contributing to the regeneration of NAD during hypoxia. How glutamine can work on this? So this here is called the malate aspartate shuttle. So basically it's the way that the cell has to move NAD or NADH across the mitochondrial membrane. If we start in the mitochondria, but the oxidation of malate into oxalacetate, this will consume NAD in the mitochondrial matrix. Then through transaminations and transport aspartate to the cytoplasm and getting the same reaction in the opposite way, we effectively generate NAD in the cytoplasmic side of the mitochondrial membrane. Glutamine can, at least at first, contribute here through glutamate and push the shuttle by pushing the transamination of glutamate and aspartate. So this is what we did. We said, okay, let's see how the glutamine contributes to this pathway. We used labeled glutamine with carbon-13 that allows us by mass spec to track where the carbons on the glutamine ends. This was actually really challenging because we need to keep the mouse, of course, alive. And for 5 hours doing a continuous infusion by IV of the tracer. However, at 7% hypoxia, it was really hard keeping the mouse anesthetized and alive. They were dying about 30 to 1 hour starting the experiment. We tried higher oxygen levels, but unfortunately at those levels, the hypoxia wasn't induced proliferation and this metabolic adaptation weren't happening. So finally, we found this solution. It's called vascular access buttons. So they are implanted in the back of the mouse, and through here, we connect a catheter that goes into the jugular vein. So we can connect in this cantilever that has the tubing lines. The mouse can be awake. This allows the free movement of the mouse in the cage, and we connect it to a pump that regulates the flow and the length of the infusion. So basically, the mouse is doing almost normal life, just under hypoxia, while we are infusing the uniformly labeled glutamine. So the first thing we found is there is no changes in the incorporation of the total labeled glutamine in the heart. This is important because if we see any change later, means that this is not due to an increased incorporation of labeled glutamine, and this is only the label fraction, the uniform label fraction into the heart. Of course, we still see the same adaptation that we see in previous experiments. When we quantify the overall pool size, the overall levels of glutamine in these hearts under infusion, there is still an increase in glutamine. Of course, it's important to mention that here we use physiological concentrations of glutamine. We don't want to push the system by infusing extra glutamine and creating an artificial increase in the glutamine concentrations in the blood. So how can glutamine contribute to the NAD generation in the cytoplasm by the malate aspartate shuttle? We start with glutamine, it's called N+5, meaning that it's the same mass than glutamine plus 5 units more because the 5 carbons are labeled. We go to glutamate, and actually here we start to see that there is an increased conversion of glutamine into glutamate in the hypoxic hearts. Then we go to alpha-ketoglutarate, enter the Krebs cycle, like classic biochemistry. We start turning these carbons around the cell cycle, the Krebs cycle, sorry. So in the first turn, all the species are labeled N+4. And actually we see an increased labeling in N+4 species of the Krebs cycle during this first turn. Of course, after 5 hours, we are in a steady state, so the carbons has been rolling here for a while, so the main labeling we see is the second turn of the Krebs cycle, that is N+2, because we are getting unlabeled carbons here through acetyl-CoA. So all the N+2 species also show an increase, a significant increase in hypoxia over normoxia, indicating a bigger contribution of glutamine-derived carbons into the Krebs cycle in chronic hypoxia. The way that this connects to the malate aspartate shuttle is at the level of the oxalacetate. If we transaminate oxalacetate, we get aspartate, N+4 or N+2, depending which oxalacetate are we getting. And we also see an increase in this labeling in hypoxia. This wasn't changing the overall levels of aspartate. The pool size is the same, indicating that there is no accumulation of aspartate. This aspartate is going through, hopefully, this pathway. Actually, the flux through the malate aspartate shuttle is controlled mainly by the GOT1 activity. This is a glutamine transaminase that catalyzes this step here. And we see that there is an increased expression by RNA-seq in the chronic hypoxic heart compared to normoxia. But one step further, and this would be the last big piece of data, is glutamine can also contribute through a cytoplasmic branch. This is really interesting because in hypoxia, we are talking all the time mitochondria is not that much active, so if we still rely on Krebs cycle, and we are actually accumulating NADH in the mitochondrial matrix, this is not donating as efficiently to the electron transport chains. Maybe Krebs cycle is not going as a rate that this can still be working and supporting glycolysis. So there is a cytoplasmic pathway that ends up also contributing to the malate aspartate shuttle and at the end to NAD generation and glycolytic flux. So if we start again with glutamine N+5, we go to glutamate, to alpha-ketoglutarate, and this alpha-ketoglutarate, instead of going to Krebs cycle, gets pumped out of the mitochondria. There, we transform it into isocitrate and citrate that would be N+5, not N+4 as before. So we can distinguish by the tracing which species is coming from which pathway. And also, this will go to oxalacetate N+3, malate N+3, and since this is almost an equilibrium, aspartate N+3. What we see is, again, there is an increase in hypoxia in the contribution of glutamine to, through all this pathway. In terms of the regulation of the pathway, the step catalyzed here is catalyzed by isocitrate dehydrogenase, and we have several isoforms depending if we are talking about mitochondria or cytoplasm. So when we check the expression by RNA-seq, we see that there is an increase in the expression of IDH1, that is actually the cytoplasmic form, and a reduction of one of the regulatory subunits of the mitochondrial isoforms. So overall, we also see an increased contribution to the shuttle through reductive carboxylation of glutamine, that is the biochemical name of this pathway, and a switch of IDH isoforms from mitochondrial to cytoplasmic during the chronic hypoxia when we are actively increasing cardiomyocyte proliferation. So the conclusions of this part is like, of course, cardiac levels of cardiac lactate levels, and I showed at the beginning, are not increased during the hypoxia-induced cardiomyocyte proliferation, but the glutamine levels are increased during this process. Second, glutamine supplementation decreases the NADH levels during hypoxia in vitro, indicating a contribution of glutamine to NAD regenerating pathways. And also, with this tracing experiment, we have determined that glutamine contributes to cytoplasmic NAD generation by the malate aspartate shuttle through 2 mechanisms. One, through Krebs cycle incorporation, classic oxidation of an aplerotic supply of amino acid carbons, or through the reductive carboxylation of glutamine in the cytoplasm. Overall, we will be feeding this step, converting, generating malate, regenerating NAD that will lead the ensurement of the ATP production by the glycolysis while we are in hypoxia. What's next? Because this was very interesting, but how we connect this with cardiomyocyte proliferation. So we are generating GOT1 cardiac-specific deletions to check the contribution of glutamine through Krebs cycle, and also IDH1 deletion, cardiac-specific, to check the role of reductive carboxylation. What do we want to check here? Of course, we want to check the NAD plus production, and here the compartment specificity is really important. Maybe the overall levels are the same, but we need to see an increase in the cytoplasm over the mitochondria. For this, we have NAD fluorescence sensors that we can put in cells or infect tissue slides and put under the microscope. So basically, we can quantify the levels of NAD in the cytoplasm or in the mitochondria by fluorescence microscopy. Also, we will perform the same glutamine label experiment, glycolysis rate and lactate production, and of course, seeing how these knockouts affect the proliferation induced by the hypoxia. The ultimate goal would be, can we get a pathway here that is controlling directly the cardiomyocyte proliferation, and can we enhance this pathway in normoxia to see if we can induce the normoxic proliferation of cardiomyocyte just by controlling glutamine metabolism? So hopefully for Japan meeting, we have an answer on that. And at this point, I just would like to thank all SADC lab, especially Hesham. I'm like honored to work with him, and he trusts a lot in all what we do in the lab. So thank you, Hesham, and all the people. My collaborator, Ashley Solmonson, is like the most talented postdoc I ever seen. She introduced me in the world of metabolic tracing, and she has been always supportive with my 10 daily emails asking them questions. Sweda Lab, also at UT Southwestern, helping with all the NADH measurement and anything related to mitochondria. And our collaborator in this project, Mike Kinter, that he has a really nice setup for targeted proteomics, so he's checking the levels of all the proteins involved in these pathways to actually have a measurement in terms of protein level and not just gene expression. And our funding, especially AHA that supports myself, and all of you for listening to me. Thank you.